Yeah. Can you hear me in the back? Is that good? All right. Well, welcome to Cardiovascular Medicine Grand Rounds. This is a very special Grand Rounds for us. As you know, we have a rich history in our division of cardiovascular medicine, and we currently have 5 named visiting professorships in honor of chiefs of cardiology who served here for a significant amount of time and had a huge impact. Last week, we inaugurated the David Pinsky visiting professorship for the first time after his 20 years of service. Our first chief of cardiology was Frank Wilson. And Frank Wilson was quite a character. He graduated from our medical school, was interested in engineering, and there was a little thing called electrocardiography just emerging. And he studied it in England and came back. And Frank Wilson really established our cardiac unit. Interestingly, Eindhoven got the Nobel Prize for developing a 3-lead EKG, but Frank Wilson developed the 12-lead EKG, which we use today. I hazard to guess that his impact on the field is about as large as anyone could imagine. So Frank Wilson, being an innovator in research, the visiting professor that our faculty vote for every year has to be an innovator. And we have just that in the presence of Joe Wu. Joe is the director of the Stanford Cardiovascular Institute. He's the Sturzer Professor of Medicine and Radiology. He went to Yale for his MD. His Ph.D. was at UCLA. He's interested in cardiovascular genomics and induced pluripotent stem cells. The main goals of his laboratory are to understand basic disease mechanisms, the idea of accelerating drug discovery through trials in a dish, and to implement precision medicine for patients. He's received many awards, including the NIH New Innovator Award, their Transformative Award, the Presidential Early Career Award from President Obama, the HA's Distinguished Scientist Award. He is immediate past, or I guess you're current president of the AHA, immediate past president of the AHA. All of the societies that matter, the AAA, whatever, he's a member of all of those. He was a member of our National Advisory Board for a number of years. Thank you. Joe Wu is a superb scientist, and we want to welcome him as our Frank Wilson visiting professor.
So thank you so much, Kim, for this wonderful introduction, and also thank you for inviting me. In honor of Dr. Frank Wilson, I had the opportunity to read up about him last night, and it's just amazing of the contribution that he's given to cardiology as an entire field. I also want to thank the invitation to come here, because it's really great to see a lot of friends and colleagues who I've known over the years. And as you mentioned, I was on the Franco advisory panel to see the growth and also the evolution and the development of this great institution here. And so again, I'm very humbled to be giving this lecture. And so my talk is on essentially kind of a summary of what we've been working on for the past probably about past 20 years. And the title here says Stem Cells and Genomics from Precision Medicine to Clinical Trials in DISH. And I'll explain what we mean by the clinical trials in DISH. And this is my disclosure slide right here. And so since this is a clinical cardiology audience here, I just want to start by giving you a background of a patient I saw at Stanford. And this is let me just change this here to laser point. This is a 57-year-old man previously seen in our clinic for newly diagnosed non-ischemic cardiomyopathy, found to have severely reduced LVEF, and then family history remarkable Okay, good. Family history remarkable for multiple sudden cardiac death, heart failure, and then because of the multiple death, prompted clinical genetic testing, and the genetic panel shows a pathogenic variant in lamin AC. So the question is, how do you, what is the mechanism of this particular disease? How does this genetic information affect its management? And how do we treat it outside of standard care? So you must have seen this type of patients before in your cardiology clinic. And again, this is somebody that we saw in our clinic. So talking about precision medicine, I think as many of you know, this is concept was first promoted by President Obama back in 2015. And much of this is made possible by recent advances in next generation sequencing, bioinformatics, and big data. And the idea is that by combining our genes along with our lifestyle and environment, we can then better predict which treatments might work for our patients. In my opinion, I think there are three key technologies that the physicians and the clinicians and the scientists should be aware of that marries precision medicine with basic research. The first key technology is really the next generation sequencing. And this technology has become much faster, cheaper, with lower error rate and broader coverage, as shown here. For example, back in 2000, it'll cost you about $3 billion in 13 years for the Human Genome Project to complete the very first human reference genome. Today, you could get your DNA sequence for less than $1,000, and turnaround time will be 3 to 4 weeks. And I have my whole genome sequence. It cost me actually around $600, and turnaround time was about 2 weeks. And so in my opinion, the second key technology is the various types of genome editing technologies, starting first with zinc finger nucleases, and then TALEN, and then CRISPR-Cas9. This was discovered by Emmanuel Charpentier and Jennifer Doudna around 2011. So now using these genetic scissors, including more recently base editing and prime editing, you can now change the DNA of animals, plants, microorganisms, and stem cells with extremely high precision. And then just this past December, the FDA approved its first CRISPR-based gene therapy for treatment of patients with the sickle cell disease. I was actually on the FDA Cell Tissue and Gene Therapy Advisory Panel, and we looked at the data. It was very promising, presented by Vertex Pharmaceuticals, and therefore we voted for the approval of Cascovy. And then the third key technology is really the iPSC, or induced pluripotent stem cell technique, and this was discovered by Shinya Yamanaka around 2006. Because these iPSCs are genetically identical to the patients, you now have a test bed to validate the human genetic mutations and variants that you identify with the next generation sequencing using CRISPR editing, that, you know, the two techniques or platforms that I mentioned earlier. In addition, you can now differentiate these iPSCs into cardiac myocytes, cardiac fibroblasts, smooth muscle cells, endothelial cells, and cardiac pericytes, what I call the big 5 cell types in a human heart. And so this slide briefly highlights the different differentiation protocols for the cardiovascular cell subtypes. So for example, you can differentiate iPSCs into mesoderm by GSK inhibition. You can then differentiate the cardiovascular progenitor cells by Wnt inhibition, into cardiomyocytes by Wnt activation, and into actually that's in the cardiac fibroblasts by TGF-beta inhibition, and then also into smooth muscle cells by PDGF activation, as well as into endothelial cells by VEGF, as shown in this cartoon here. And so over the past 10 years, my lab at Stanford, and also more recently in collaboration with the startup company Greenstone Biosciences, we have made iPSCs for more than 2,500 patients with different medical history, ethnicity, and sex. This is the world's largest biobank of human iPSC lines. These lines are then linked to medical information. We also perform multi-omic sequencing to help us understand how human genetic variants impact drug response. We also work with the FDA on drug safety testing, and then we share this commonly with the entire scientific community. And today, we've actually sent out more than 4,000 vials to more than 400 different investigators throughout the country. So showing here some examples of cardiac diseases, as well as their genetic variants, and then as well as examples of non-cardiac diseases and genetic variants. And again, we make these cell lines freely available. So if anybody in this audience wants to sell, so we just ship it to you. We don't ask for authorship, and you just pay for the shipment costs, and that's it. Yeah. And so in terms of the R&D pipeline, this is our typical workflow. We recruit from diverse patient population in our clinic. We then generate their 2D micropattern human cardiomyocytes, as well as the 3D human cardiac organoids, and also the 3D engineered heart tissues of disease modeling and drug discovery. And then we can also generate the fibroblasts for fibrosis-related projects. We can generate the vascular cells for inflammation-related projects, liver cells for metabolism-related projects, excuse me, and other cell types as well. And so in terms of deciding whether to go with 2D versus 3D organoids versus 3D engineered heart tissues, as well as animal models, it really depends on your experimental design purpose. And so as expected, there's pros and cons with each one of these models. So your job as a scientist is really to figure out what is the exact question that you're asking, at which step of the screening process will you use which technique. And so as shown here, for example, the 2D cardiomyocytes will give you much higher throughput, but much lower maturation, whereas a 3D culture will give you much higher tissue maturation, but lower throughput and also more variability. So there's no one size that fits all. And so taking all these into consideration, this is our typical drug discovery workflow that we do at Stanford, which goes typically from 2D to 3D to animal models. Here is an example. I showed an iPSL reporter line, in this case with ACTA2 driving clover. These cells are cultured in 2D monolayer and then differentiated into, say, cardiac fibroblasts. We then screen for, say, 5,000 compounds at 7 dose concentration. The EC50 or the TC50, these are effective dose and then toxicity dose. They can then be calculated based on the dose-dependent curves of CLOVER2 and also the host sustain. And at step 5 here, the initial candidates or the filter top 10 compounds with the satisfactory absorption, distribution, metabolism, excretion, and toxicity profile. The top 20 candidates can then be undergo counter screening in iPSL-derived cardiomyocytes and endothelial cells, just to make sure you don't have any off-target effect. And afterwards, at step 6, the anti-fibrotic effect of your top hits can then be validated in additional multiple iPSL lines, as well as 3D engineered heart tissues. And at step 7, the therapeutic effect of the top hit can then be examined in different mouse models of cardiac fibrosis. And then finally, in step 8, the molecular target of your lead compound can then be identified by surface plasma resonance, by in silico docking, by molecular dynamic simulation, and also by single cell sequencing. So this is an example of a typical workflow that we do in our lab. So with these background slides, I just want to give you examples of how we use the different tools of genetics, epigenetics, genome editing, stem cell biology, and to elucidate the mechanisms of various diseases in cardiovascular medicine. For example, coronary artery disease, aortopathy, cardiac arrhythmia, genetic cardiomyopathy, pharmacogenomics, and so forth. And so the first example I'll show you is a familial dilated cardiomyopathy, or DCM. This is one of the most common causes of heart failure and heart transplant. And so in this study published about 12 years ago, we generated iPS cells from a 3-generation family with DCM. In fact, the proband here was a 15-year-old boy who was referred to us. He already had a heart transplant, DNA sequencing shows that it's due to a point mutation in cardiac troponin T with arginine to tryptophan switch and codon 173. And then compare to the healthy control within the same family, we found that these DCM cardiomyocytes are altered regulation of the calcium ion, decreased contractility, and also abnormal distribution of the sarcomere alpha actinin. And you could treat these cells with the drugs such as beta blocker, metoprolol, to improve the function of these DCM cardiomyocytes, recapitulating results from multiple large beta blocker clinical trials. And another example, we will refer a large family with familial hypertrophic cardiomyopathy, or HCM. This occurs in about 1 in 500 patients, as you know. In this case here, it's due to a myosin heavy chain 7 mutation with arginine to histidine switch at codon 663. The mother here had HCM with symptoms of cardiac arrhythmia, and then she has 8 kids. The 2 oldest are already manifested the HCM phenotype, whereas kid number 3 and kid number 8, they were both genotype positive, but phenotype negative, most likely because they were still young at that time. And so through a whole series of experiments, we're able to understand mechanistically how this particular myosin heavy chain 7 mutation increases intracellular calcium, which leads to calcium handling defects and the cellular arrhythmia. It also causes activation of the calcineurin infrax signaling pathway, which leads to hypertrophic responses. And again, we use this in vitro setup for drug screening, and we're able to show that a calcium channel blocker, such as verapamil, shown here, and beta blockers, such as propranolol, can be used to reverse both the cellular hypertrophy and the cellular arrhythmia. Now, in the pediatric population, the third most common type of cardiomyopathy is left ventricular non-compaction, or LVNC, and it accounts for about 1 in about 1,000 patients. So this is another study in which we will refer this large family here from Stanford and a smaller family cohort from Japan. The boy here at Stanford already had a heart transplant. His older brother, older sister also had LVNC, but were asymptomatic. And interestingly, his father had mild dilated cardiomyopathy without LVNC, and his paternal grandmother had cardiac arrhythmia that required pacemaker. And so through a whole series of experiments, we're able to confirm that the mutations in cardiac transcription factor, TBX20, one in the T-box domain for the Stanford boy, and then the other one in transactivating domain for the Japanese girl, actually causes abnormal activation of the TGA beta signaling pathway, which recapitulates a key aspect of the LVNC pathophysiology. And this is actually the explanted heart of this Stanford boy, as shown here. Now, so far, what I've shown you are like well-known examples of 3 Mendelian genetics with strong phenotypes causing genetic cardiomyopathies such as DCM, SCM, LVNC. But as you all know, the human genetics is much more messy, much more complicated, especially when you're dealing with the variants of unknown significance, or VUS. This is a real clinical conundrum for doctors like us who are taking care of these patients. So for example, showing here is a proband who is a healthy middle-aged man who underwent DNA sequencing as a part of an executive VIP workup as a concierge clinic. He was found to have a likely pathogenic variant in myosin light chain 3 with cysteine to alanine switch at codon 170, along with multiple family members, his mother, brother, and also daughter. So the clinical dilemma is, what do you do with this healthy individual? DNA sequencing shows possible pathogenic VUS. Well, you could wait it out for the next 10 years and see more papers describe this particular variant. Or you could try iPS cells and genome editing to assess the functionality of this variant. This patient was actually referred to our clinic or our lab. And so here, we first generated iPS cells from VUS members and isogenic controls and showed that their cardiomyocytes display normal phenotype at baseline. Second, when we introduce a homozygous variant by CRISPR genome editing, meaning that the patient now has 2 copies of this potentially pathogenic variant, his cardiomyocytes remain normal. Third, when we disrupted the healthy allele by introducing a frame shift mutation, the cardiomyocytes were also normal, meaning that what was annotated as a potentially pathogenic variant allele was still able to perform a normal function. And then finally, just to make sure our assay is working correctly, when we genome edited a well-known pathogenic variant in myosin light chain 3, his cardiomyocytes start showing severe arrhythmic phenotypes. So based on these 4 steps, and based on the fact that the patient himself was clinically healthy, we concluded that what was initially annotated as a likely pathogenic variant is wrong, and it's actually most likely a benign variant. And actually, ClinVar, which did the initial annotation, now included our study into their interpretation as a potentially benign variant instead. Now, previously, I've shown you that the iPS cells can be used to model mandated mutations, as well as model variant of unknown significance. In addition, they can also be used to study impact of single nucleotide polymorphism. And I think the most common example I want to show is the aldehyde dehydrogenase, a genetic polymorphism. I think some of you may or may not know that the common Asian flush syndrome is actually due to a genetic variant in the aldehyde dehydrogenase. It is one of the most common genetic polymorphism in the world. This enzyme deficiency is present in about 36% among East Asians and 8% of the world population. It causes alcohol flush reaction, which, when you drink after 10, 20 minutes, there'll be flushing, nausea, tachycardia, and headache. And actually, I'm missing this particular enzyme. That's why we decided to study this. And my face looks exactly like this after 10, 15 minutes when I drink a glass of alcohol. Now, interestingly, the aldehyde dehydrogenase 2 polymorphism with glutamic acid, the lysine switch, this has been linked to increased risk of coronary artery disease, more severe outcome after heart attack, higher incidence of hypertension and cancer, and increased complications of type 2 diabetes. So this piqued my interest in terms of using iPS cells to study the impact of this genetic polymorphism. And so, as I mentioned earlier, we know from large genome-wide study, GWAS study, using the Biopink Japan data set, that this enzyme deficiency is associated with CAD. So here we differentiated iPS cells into endothelial cells from these patients to model vascular dysfunction after alcohol exposure. We also corrected these deficient cells into normal phenotype cells by CRISPR editing. We also did compound library screening and molecular docking analysis. And in fact, we showed that SGLT2 inhibitor, empagliflozin, can actually mitigate this vascular dysfunction. And these findings were also confirmed in mouse models of diabetes and alcohol exposure, showing that empagliflozin can actually ameliorate endothelial dysfunction in both disease conditions. So that was an example using iPS cells to study alcohol intolerance among East Asians. But you should be able to use this platform to study other genetic susceptibility among different ethnicities. For example, we're interested in understanding why African Americans have much more hypertension, why Hispanics have much more diabetic vasculopathy, and also why South Asians, for example, Indians and Pakistanians, have much more metabolic syndrome. So I want to switch gear now to the second part of my talk, which is using these iPS cells for drug discovery. And I think, as you know, this is a very time-consuming, expensive process. For example, typically it'll cost you about $1 to $2 billion in about 10 to 15 years to bring a new drug from discovery to clinical trials to market approval. And in the US, both the FDA and Big Pharma are also thinking about using these iPS cells to accelerate the drug development process. This is a study published by several companies and also by the FDA, in which they tested human iPS cell-derived cardiomyocytes to predict cardiac arrhythmia risk as part of this CIPA initiative, or Comprehensive In Vitro Proarrhythmia Assay Initiative. And here, the investigators used one consensus protocol, 2 human iPS lines, 3 continents, 28 blinded drugs, 2 statistical models, and 10 volunteer labs. Importantly, they were able to show 87% prediction rate. This is very good considering that this is actually one of the earliest studies by the FDA group, and therefore there's going to be significant room for improvement in the future. And in our lab, we've also been pushing that direction as well. This is a paper we published in 2016 using transcriptomic profiling of iPSL-derived cardiomyocyte to predict individual drug safety and efficacy responses in vitro. Here, we tested 2 drugs, rosiglitazone and tacrolimus. We also used CRISPR genome editing to evaluate and confirm the potential mechanisms of action, as well as the adverse effects. In particular, we focus on the mechanisms of rosiglitazone. This is a PPAR gamma agonist that was used for diabetic patients in 2000, but was pulled off the market in 2010 because of the increased cardiovascular death. And because of time, I won't go over the details of these experiment results. So one of the goals in our lab is to start creating a human atlas. A human atlas has drug transcriptomic signature using iPSL heart tissues. For example, I take Lipitor every day. It checks. But what is the effect of Lipitor on my heart muscle, on my liver, on my brain? We don't know. And because my primary doctor is only able to check the LDL. And so here, this is an area of interest to us. And I just showed 2 examples whereby we assess the effects of 4 different kinds of calcium channel blocker, nifedipine, amlodipine, diltiazem, and verapamil, 4 different kinds of statins, atorvastatin, simvastatin, lovastatin, and fluvastatin. And what is interesting to us is that you will see different gene expression patterns among different drugs in the same patient, as well as different gene expression patterns among the same drug in different patients. And so this suggests that even though they belong to the same class of drugs in general, these drugs may exert very, very different effects among the different individuals. Now, besides cardiology drugs, we also have an interest in studying chemotherapy drugs. And that's because heart disease and cancer are the top 2 causes of morbidity and mortality in the US. And as more cancer patients survive due to better immunotherapy and chemotherapy, you're going to have more heart failure symptoms due to cardiotoxicity from these drugs. And so in one study, we used human iPS cells from breast cancer patients to predict doxorubicin-induced cardiotoxicity. I think, as you know, doxorubicin, this is one of the most common chemotherapy drugs that are given to breast cancer patients, but it can cause cardiotoxicity in about 10% of the patients. The problem for us as clinicians is that we don't know which one of these 10% will develop as doxorubicin-induced cardiotoxicity. And so here we recruited breast cancer patients. A year later, some of them had normal cardiac function, while others had doxorubicin-induced cardiomyopathy. We generated the iPSL-derived cardiomyocytes, exposed them to doxorubicin in vitro, and then studied the cellular responses by using these assays as listed here. And then here, as proof of principle, we showed that if we pick the patients appropriately, these iPS cells, essentially you're using them as a mini chip readout, can be used to recapitulate which patients are more susceptible to doxorubicin-induced cardiotoxicity. So another popular class of chemotherapy drugs are tyrosine kinase inhibitors, or TKIs. And TKIs kill cancer cells by inhibiting the phosphorylation activity of hyperactive receptor tyrosine kinases. Common TKIs you may have heard of include Gleevec and Sutent, which inhibit VEGF-R2 and also PDGFRB in cancer cells, but also cause heart failure as a side effect. And this is because both VEGF and PDGF signaling pathways are also critical regulators of cardiovascular development. And so here we generated iPS cells in 13 patients, treated them with 21 most common TKIs, and came up with what we call a cardiac safety index that allows you to predict which TKI is cardiotoxic and which TKI is not. And so our ultimate goal is to further optimize this cardiac safety index so that if you give us a TKI, we can help you predict whether the drug is toxic or not before you further develop this. Now, these days, you cannot give a talk without saying something about AI machine learning because of its broad impact in biomedical science. For example, you can use AI to interpret EKG. You can use AI to assess LVEF and echo MRI, and also to monitor cardiac arrhythmias using wearable devices. At the same time, there's also significant advances in drug space using AI. As you all know, the Nobel Prize for Chemistry was given for somebody who invented the AlphaFold, right, which is a common AI algorithm to assess a protein structure. Now, working in collaboration with computational scientists at Greenstone, we routinely use these AI softwares that allows us to analyze the protein structure by in silico molecular docking. And once we know the molecular target and the protein structure of the disease we're going after, we can actually do virtual compound screening for about 1 billion compounds. And these are compounds that have never been synthesized before. It'll take us about 6 to 8 weeks to screen through all these 1 billion compounds. Once we screen them, we can then synthesize these compounds. And so this is a schematic of our typical molecular docking campaign. You can actually, besides the virtual library, you can also start with the existing library, such as Zinc 22 library, the FDA repurposed library, the Enamine library from Ukraine, also the Wuxi library. Or you can start with the even smaller library. For example, at Stanford, we have a 100,000 compound library. And once you identify the molecular target, the molecular docking site that explains the mechanism of action for your disease, you can then further refine the rank score as well as perform autodock and molecular dynamic simulation to look for compounds with the desired drug-like properties. So here I just want to highlight a couple of examples of using AI machine learning for in silico docking for drug discovery. One example is marijuana. I think, as you know, marijuana is being used increasingly in the US due to legalization. In California, it's legal. I'm actually not sure what it is in Michigan. About 35 states are legal right now. And when we look at the UK Biobank data set, we actually found that marijuana use is associated with increased myocardial infarction, but the mechanism is unknown. And here we worked out the mechanism that the delta-9 THC, this is a psychedelic component of marijuana. It can bind to a cannabinoid 1 receptor and cause vascular inflammation and oxidative stress by activating MAP kinase, NF-kappa B, and cytokine pathways, which then causes oxidative stress, inflammation, and cell death. So we reasoned that this is our target, and using AI machine learning and also in silico chemical library, we actually screen for a drug, and it's a Genestin, as shown here. It serves as a neutral CB1 antagonist, which means it can competitively block the binding of THC. It sits there, but it doesn't activate it, and therefore it prevents more THC from binding to the CB1. And Genestin, interestingly, is a flavonoid that's found abundantly in soybean. And so my former postdoc, Mark Tandy, who's now a faculty in Canada, he's actually doing a clinical study right now by giving Genestin to HIV patients because HIV patients typically have high vascular inflammation. Now, here's another study using iPS cells for and also genomics and AI machine learning for drug discovery. We know cardiac fibrosis is bad because it impairs cardiac function, but there's no effective clinical therapies. So using these high throughput assays as well as iPSL reporter line, we identified artesunate as a potential drug that we can repurpose. Artesunate is a derivative of artemisinin and dihydroartemisinin, which are the primary drugs that are used to treat malaria and it's been given to millions and millions of people. So the Chinese scientists who discovered artemisinin, Dr. Tu Yu, actually received the Nobel Prize in 2015 in medicine for this discovery, and useful as well as in vivo mouse model. Mechanistically, we showed that the TGF-beta can stimulate the fibrotic response by canonical SMAD and also the non-canonical ERK pathway, which regulates these extracellular matrix proteins. And these extracellular matrix proteins can stimulate the MD2-TRL4, which then further activate the whole cascade. And so we use the in silico simulation, and we're looking for drugs that can actually block this MD2-TRL4 interaction. And again, we found artesunate as a very potent compound and did all the subsequent analysis to show how this works. Now, for the last part of my talk, I just want to touch upon how we can then use this human iPS cell to implement the concept of clinical trial in a dish. Here, the concept is that we should be able to recruit patients, generated the different cell types of interest, test which patients respond to drugs and which patients do not, and then go back to perform the actual clinical trial on patients who are positive responders. And that way you have a higher chance of a success rate for phase 1-2 clinical trials, decrease the cost, and also accelerate the timeline for drug development. I think this concept is quite timely because, as some of you may know, the FDA Modernization Act 2.0, this was passed by both the Senate and the House on December 2022. So just think about when was the last time both our Senate and House agree on something unanimously. Now, it's pretty rare, right? So this law authorizes the FDA to use alternative testing platforms such as stem cells, organoids, and in silico modeling. It's just like what I've been showing you. And for some drugs to completely eliminate the requirement of animal testing before entering clinical studies. And the goal here is to improve the predictiveness of preclinical safety and efficacy assessments. Now, as I mentioned earlier, I'm on the FDA Cell Tissue and Gene Therapy Advisory Panel. And this slide is actually given to me by my colleagues from the FDA. For rare orphan diseases, the FDA recognizes the challenges of developing new drugs due to small number of patients, and yet there are thousands of genetic variants for each disease. So now their approach is to accept data generated from cell models to test the drug efficacy instead. So as an example, cystic fibrosis, the drug Callidico, made by Vertex Pharmaceutical, initially approved for 10 genetic variants, now has been expanded to 24 more genetic variants based on cell model. Another example is Fabry's disease. This is a lysosomal storage disease passed on by the X chromosome. The drug Galafold, made by Amicus Therapeutics, initially approved for 40 genetic variants and now has been expanded to 348 genetic variants based on cell model. And so currently the FDA is very interested in expanding this cell-based platform across the full spectrum of diseases. And so this slide is also given to me by the FDA. I think the value proposition here is using these in vitro efficacy data to reduce the number of clinical studies, to inform the regulatory decision-making at the time of marketing application, to streamline the drug development process, and also to expand the indications based on your in vitro data. This slide highlights, in my opinion, what I think is one of the major problems for the high failure rate in drug discovery, and that is the lack of genetic diversity and pharmacogenomic differences with traditional models such as immortalized cell line or genetic inbred mouse model. For example, because these are immortalized and inbred, the deviations that you see here within this group versus this group, the deviation here is actually due to your technician's pipetting skill for cell culture work. The deviations that you see here in the animal work right here, this group versus this group, is actually due to your technician's tail vein injection skill, right? Because these are all just inbred, it's exactly the same. On the other hand, the differences that you see in the population of, say, 1,000 diverse iPS cell lines is due to underlying genetic variability that exists among all of us. And this pharmacogenomic difference is ultimately what you're trying to understand before you go into the actual clinical trial that is otherwise impossible to capture, recapitulate using traditional immortalized cell line inbred mouse cell models. So I think the FDA Modernization Act will actually pave the way for the next era of drug discovery that uses predictive AI or generative AI, similar to what I've shown you with all these in silico docking and also with AlphaFold, to find the optimal candidates, similar to the story of Genestin as a neutral CB1 antagonist, and also artesunate to bind the MD2-TRL4 pathway. And then it doesn't matter whether these are drugs, peptides, antisense oligonucleotides, and so forth. You do your in vitro and studies and also your in vivo traditional PK/PD studies. And then afterwards, a clinical trial in a dish model can then be used to help you predict the safety and efficacy of your lead compound before you test them in larger clinical trials. So I just want to wrap up my talk by going back to this clinical case report. As you'll recall, 57-year-old man referred to Stanford Clinic, multiple family members died from sudden cardiac death, reduced EF, heart failure, actually hypertension. And that was quite interesting as well, because usually low EF is associated with low blood pressure. So it turns out he belongs to a very large family cohort with Lamin HC frame shift mutation that leads to early termination. Lamin cardiomyopathy accounts for about 10% of familial dilated cardiomyopathies. It is the second most common type of genetic cardiomyopathy, with titin mutation being the first one. And as you can see here in this large family, multiple individuals had cardiomyopathy, atrial fibrillation, AV block, and as I mentioned, 8 of them died from sudden cardiac death. So this is a horrible disease on this particular family. Here, mechanistically, we found out that abnormal activation of PDGF in their cardiomyocytes can cause the abnormal cell arrhythmia and also the impaired contractility as shown in this cartoon. And again, because of time, I won't go over all the details. We also showed that by blocking PDGF pathway with the PDGF inhibitor, quinolonib, a very low dosage can rescue the abnormal phenotype of these cardiomyocytes. When we look into quinolonib, the problem is that it was a Pfizer drug. They abandoned it. They gave it to a company called AROC in Texas. AROC is in phase 3 trial for AML, ALL at a much higher dose. And they were interested in partnering with us for testing this drug in our patient because they don't want to, quote unquote, rock the boat. And so we didn't pursue quinolonib as a potential PDGF inhibitor for these patients. But what was interesting to us is that, as I mentioned, many of these patients had high blood pressure and at age 30s and 40s. And we were quite puzzled by this clinical finding because, as I mentioned, typically the blood pressure is lower in patients with dilated cardiomyopathy. And so here we decided to differentiate this family cohort's iPS cells into vascular endothelial cells and then compare the difference between Lamin versus the healthy control. And to make a long story short, KRF2 is a transcription factor that is responsible for the endothelial dysfunction. And here we use a drug library screen looking for FDA-approved compounds. We found that lovastatin can activate KRF2 and rescue the endothelial dysfunction. And then here's the mechanism that we worked out. In contrast, the lovastatin is, as you know, FDA-approved. So we can then repurpose this for these patients. And that's what we did. And hence the title here is Clinical Trial in a Dish using iPS cells showing lovastatin can improve endothelial dysfunction. So the same Lamin patients, when we tested the endothelial cells, were asked to return to our clinic, treated with the lovastatin daily, even though they had normal cholesterol numbers. We're not treating it for cholesterol. We're treating for vascular dysfunction. And the clinical data is shown here. We use EndoPAD, which is a machine that can assess flow-mediated dilatation during reactive hyperemia. Normal index is considered 1.67 and above. This is essentially a blood pressure cuff. You occlude it for 5 minutes, you release it, and see how much blood flow comes back. As you can see here, this Lamin patient number 2 started with a very poor index of 0.48, and it improved significantly to 1.06 after 6 months of lovastatin treatment. So I think this is a clear example of what we call a clinical trial in a dish, whereby we first identify a large family with Lamin mutation, elucidated the mechanism of vascular dysfunction, screened for a panel of FDA-approved drugs using their iPS cells, found that lovastatin can be used to improve the cellular phenotype by enhancing the KRF2 pathway, confirmed the in vitro findings in the actual patients themselves by using EndoPAD as a surrogate readout after 6 months of lovastatin treatment. So in summary, I hope I have shown you the framework of how there may be a paradigm shift in the future of involving using patient iPS cells to help us better understand first the molecular mechanisms of action, the biological specimen, what I call the biological data, can then be converted into digital data with the multi-omic sequencing pathway analysis, drug target profile of your cardiomyocytes, your endothelial cells, your fibroblasts, and so forth. These digital data can then be fed into AI machine learning softwares for molecular docking, molecular dynamic simulation, in silico admin prediction. This will then give us drug targets that are much more precise and predictive. The lead compounds can then be tested on their patient-specific iPS cells that have the desired disease phenotype that you designed the drugs for, and hence completing this loop of a clinical trial in a dish. And the ultimate goal is to decrease the cost of drug development, shorten the timeline, and develop therapeutics that are much more efficacious and safe for our patient, which in my opinion is really the holy grail of precision medicine. So this concludes my talk. I want to acknowledge my postdocs, my graduate students, instructors, and my former postdocs whose work I highlighted, and also my the funding support from the NIH, from the AHA, Chen Zuckerberg FDA, and also in collaboration with Greenstone Biosciences. And as I mentioned earlier, we have a collaboration with Greenstone Biosciences in which they can actually give you iPS cells to academic sites are free of cost. There's no charge, no authorship required. You just pay for the shipping costs. And these are iPS cell lines from cardiac and non-cardiac diseases. And also these are the different universities, just a sample list of different schools that have received cells from us. Our goal is really democratize the whole system so that the more people who study iPS cells, the faster the field moves as a whole. And for those of you who's interested in drug discovery, at Stanford, I organize an annual drug discovery symposium. The one in 2025 will be on April 28th and 29th. And typically here we bring in key stakeholders, FDA, big pharma executives, CEOs, CSOs, CMOs, and then biotech, as well as venture capital investors, as well as NIH and academia. 2-day symposium. We talk about how do we accelerate drug development. This is free if you join this virtually, and then we also have this in person. And with that, thank you very much for your attention. I'll be happy to answer any questions. Yeah.
Thank you, Joe. Wonderful presentation. Tony.
Thanks so much. That was a real remarkable tour de force and a remarkable body of work. Thanks for sharing that. And I just want to say you have always, even before Greenstone, been remarkably generous in sharing knowledge, reagents with the community. So thank you for that. It's a great vision and it's exciting, and I think it is accelerating discovery. I wonder if you can talk a little bit about some of the future and current challenges and limitations for this. So, for example, one thing with the Lyme mutation, where we know the cardiomyocyte, and the endothelial cells are integrally involved, but even in HFpEF, for example, Craig Lewis has shown that a lot of the defect is peripheral oxygen extraction. So settings where we're not quite sure what the target cell lineage is, or where it depends on multicellular interactions that we may have trouble modeling, or perhaps environmental influences, aging, and other things that are hard to model in the dish. What are your thoughts about approaching those, which do so much of modern medicine?
So very good question. And as usual, Tony is very, very insightful with these questions. I think in general, what I would envision and hope is that the cost of doing these type of experiments go down. The expertise we basically share with everybody, the more investigator uses. It's almost like, 50 years ago when the transgenic mice came about in the beginning, very appealing, and right now everybody uses it. Our goal is that 10 years from now, 20 years from now, this is a standard platform. Everybody use these iPS cells, everybody use CRISPR editing, everybody use genome sequencing to help understand cardiovascular disease at a fundamental level. Just to give you a perspective, I started working this back in 2004 when I showed up at Stanford. At that time, just the cardiomyocyte alone, we don't do this monolayer differentiation. We were using a hang and drop assay. It's on the cover slip, you invert it, and the whole embryo body beats, and we're pumping our chest and say, look, we got something beating. And within that beating embryo body, it's only 1% to 2% cardiomyocyte. Fast forward 20 years later, I was talking to Todd today. We can get 90% plus cardiomyocyte with our eyes closed. Very easy. And this tells you how fast the cardiomyocyte field moves. Same thing with the cardiac fibroblast, same thing with the endothelial cells, same thing with the different cell types. So as more investigators work on it, the knowledge gains goes up, and then the cost goes down. And then it's a matter for us to kind of figure out how do you make it more sophisticated, how do you make it 3D, so that you understand the interaction within your engineered heart tissue or cardiac organelle. It's the interaction between your cardiomyocyte, endothelial cell, smooth muscle cell, fibroblasts. This is all possible. We could generate all 4 cell types of the NAMM patient, put it together, and do single cell RNA sequencing to see what is the crosstalk. You could take a cardiomyocyte from NAMM patient, put it with Joe Wood's fibroblasts, endothelial cells, and so forth, to see how does a diseased cardiomyocyte exert its bad effect on neighboring cell. A lot of this is possible because you could tease it out with single cell. You could also tease it out with these stuff. And with the environmental, yes, one of the drawback of iPS cells is not able to capture the epigenetics, meaning that, and the way I kind of explain it to the people who are not in the field is, let's say, for example, Kim and I are identical twins. Genetically, we're the same. Kim exercises every day, and you can see he's in perfect shape. I somehow, get lazy, get divorced, and I had a depression, and I eat hamburger diet every day for the next 10 years. I get heart attack, hypertension, diabetes, BMI 40. The iPS cell will generally look exactly the same. You won't be able to tell that because you can't capture all of that stuff. Now you say, well, that's a problem, that's a bummer. But I think you kind of have to look at it and say, okay, knowing its deficits and knowing its strength, what can we do? Let me give you an example. Let's say, for example, you're interested in epigenetics or gene expression profiles and methylation profiles. And then if I were to pass away, you would take my heart and you would do the sequencing, methylation profile, RNA-seq, ATAC-seq, whatever seq out there. And if you happen to be a diabetic researcher, you would say, aha, the difference between my heart and normal heart is all due to diabetes. If Eugene happens to be a hypertension researcher, which is, aha, all these differences are due to hypertension. If somebody else happened to be a cocaine researcher, say, aha, Joe is using cocaine, all these differences is due to cocaine. Somebody else is working on obesity, is, oh, all these are due to MASH and obesity related. So when you look at how we do stuff, when you look at a patient and biopsy them at age 60 and 70, and you say, oh, this is all due to disease, one particular disease. That can't be true, right? It's all due to amalgam of all the different disease conditions. But you know that, and now with the iPS cell, what you could do is take the iPS cell, expose it under diabetic condition, hypertensive shear stress condition, expose to cocaine, exposure to diabetes, and expose to high-fat diet condition. You could do the same ATAC-seq, RNA-seq proteomics. You could try to match it and try to help you further refine what are the signatures due to diabetes, what are the signatures due to obesity, what are the signatures due to hypertension. So you just have to look at it and say, well, it can't answer all these questions, but what is it that we could do? I gave you the example of marijuana. I mean, that's a postnatal epigenetic changes. We also, one of my postdocs is interested in studying this for microplastics, and another postdoc is interested in studying this for nicotine exposure. So I do think that it's quite powerful. The only deficit is the cost, the time, the epigenetics, and some other nuances. But this is not alchemy. This is really essentially, it's an embryological developmental thing, right? It's almost like, I explained this, it's almost like your dad's sperm, your mom's egg mixed together, and voila, after 9 months, the baby comes out. We just don't understand all the process involved. With the iPS cell, you could essentially generate all these cell types that comes out, yeah.
How do you deal with gender of the cell lines?
Yeah, so we have both males and females, and so I think you can differentiate to whatever cell types, and you can then compare the endothelial cells, the cardiomyocyte, let's say 10 male versus 10 female, 20 male, 20 female. You look at specifically at the changes, albeit a lot of times you might see very minimal change. The main reason is that if you do RNA sequencing, you're looking at a full spectrum of 20,000 genes, and the ones that are counted by XX versus XY gets hidden within that
Right.
20,000. So
Sure.
you need to zoom in. Now, you can also use this model and say, okay, I want to take the iPS cells and I want to expose it to increasing dose of estrogen, increasing dose of testosterone. I want to see what is the effect of estrogen testosterone in causing arrhythmia. I want to use this to model effect of estrogen testosterone and vascular dysfunction. You could use this to model a lot of the diseases. Yeah.
Right. Are there other questions? Yes.
Great. So sort of a follow-up, Tommy's question, do you think it's possible to, let's see, reverse aging in a brain condition?
Reverse aging. Yeah, so these iPS cells are more of an immature stage. And so what a lot of people want to do is actually push them to more mature or use this, push them to more senescent. I haven't come across people that try to take these models to reverse aging. Having said that, we have progeria models. So we have iPS cell lines from progeria patients. We differentiate the endothelial cells. We're screening the endothelial cells with the FDA-approved compound with a whole bunch of other compounds to see what drugs can reverse vascular senescence in the progeria model.
If Dr. Wilson was alive, he would be very happy to know you can do an EKG on a beating heart in the dish. I'm sure of that. Dr. Wilson.